My name is Nicole. I'm the new executive director of the American Porphyria Foundation, and I'd like to welcome you all and thank you for being here to kick off Rare Disease Week of 2024. This Zoom Good Genes Gone Bad is being presented by Dr. John Manak. Dr. Manak is now at the University of Iowa, and he is a molecular geneticist. He also has a rare disease, so he knows where a lot of us are coming from. So Dr. Manak, thank you so much for being on today. Everyone is ecstatic to hear what you're going to say, and so take it away.
So maybe I can talk a little bit and tell everybody my background. My rare disease is not particularly rare, actually. It's a common disease, but my form of it is rare. So I'm a type 1.5 diabetic. So what my lab does here at the University of Iowa is we study human genetic disorders. What we do is we obtain DNA from either large disease cohorts or families, and we basically identify the genes associated with those diseases or disorders. So we've identified new genes for spina bifida, a genomic rearrangement for a disorder called branchiotorenal syndrome. I'm particularly proud of a real key gene we discovered that was associated with bilateral renal agenesis, where kids are born with no kidney. That's not compatible with life. And then finally, we published a paper last year in 2023, where we looked at over 1,000 patients with cleft lip and or palate and discovered a number of new genes associated with that disorder. So I'm kind of well steeped in human disease genetics, and I've sort of been bringing myself up to speed with regard to the porphyrias, because this has been sort of a really fascinating topic that I've gotten interested in, partly because one of the courses I teach, the Good Genes Gone Bad class, we cover porphyria. And this is basically the holy grail of human disease genetics, because every kind of complication exists for this disorder. And hopefully by the end of this disorder, either you're going to be very happy or maybe very displeased, because there are a lot of big question marks regarding diagnosis and, prevalence of the disease, right? Why does it affect certain family members and not others? Hopefully I'll be able to cover all that information. This is a very particular disease transmission for the porphyrias. But, in looking at so first of all, looking at the comments, AIP, great. The most common hepatic porphyria, PCT, most common, the other most common porphyria, right, cutaneous. So you're well represented here. That's good. What I thought I would do to start out with, though, is just bring it down to the most basic, simple concepts. I'm going to tell you about DNA. I'm going to tell you about mutations before we get into any of the really complex stuff. Yeah, if people could please mute so that there isn't back chatter, that would be great. And, in actually looking at the questions that were sent to me, I think it really makes sense to just assume that most people on this Zoom are going to have a fairly basic rudimentary knowledge of biology and genetics. And hopefully by kind of giving you some of the basics, you'll really start to understand when you get, a genetic test, how to read it, right? Okay. So everybody can see my PowerPoint. Hopefully it's filling the entire screen. Is that correct? Okay, great. So I'm at the University of Iowa. I have an appointment in both biology and in pediatrics. And so I partner with a number of collaborators over in pediatrics to obtain the DNA samples we use in my lab. So I wanted to throw out this really interesting piece of information, because a lot of people don't appreciate what happens, how many mutations we actually sort of inherit, but that inherit isn't really exactly the right term here, from our mothers and fathers. There's the actual mutations that we directly inherit from mother's DNA or dad's DNA, but there's also DNA that is not present, mutations that are not present in the mom, mutations that are not present in the dad that wind up in the child. How can that be? Well, it's because of sort of germline development, germline meaning eggs in women, sperm in men, right? And when the process of gametogenesis happens, basically mutations can happen. So I've thrown up this statistic here. Can everybody see my pointer when I move it around? Everybody see that? My little arrow here?
No, I don't see it. I'm sorry.
Okay, let me see if I can change this so that everybody can.
There's an annotate on the bottom left. I don't know if that'll help.
That's not what I want here. Yeah, I'm looking at this annotate. Usually I have the ability to laser point.
I think maybe it isn't. It's showing the next slide, so maybe it isn't in full screen.
You can see your pointer though.
Oh, you can. Yep. Oh, there it is. Yeah.
I can see, you guys can see it. Okay, great. Good. Okay. All right, good. So the bottom line is we inherit about 100 mutations from our mother's germline and 100 from our father's germline. So that's 200 new mutations that neither of your parents had that you have, right? Now, fortunately, we have 3 billion base pairs of DNA, so the likelihood that one of those mutations is going to hit something important is relatively low, but it does happen, right? And these are called de novo mutations, de novo mutations. And sometimes when we want to sort of find potential low-hanging fruit, let's say there's a child that has a disorder, neither parent does, we look at de novo mutations, right? Because it's something different about the child that's not in the parents, just like the disease, right? So hopefully people know who this person is. This is the most popular celebrity based on Marie Claire magazine a few years ago, Selena Gomez. And if we were to kind of pull apart all of her cells in her body, there would be about 37 trillion of them, 37 trillion cells that make up a human. And this is kind of the basic schematic of a cell. And you don't have to know a lot about all the workings of the cell, other than there's a kind of a globular, kind of a circular structure in the middle of the cell that's called a nucleus. Inside the nucleus are chromosomes, and the chromosomes are basically made up of DNA and protein, and on those chromosomes are the genes, And what I'm going to talk about in a little bit is that copies of a gene from the DNA in the nucleus have to be made. And that copy is called RNA. And that RNA then gets transported from the nucleus into this goo out here called the cytoplasm. And these things called ribosomes, they're machinery that read the RNA and translate it into protein, okay? So I'm going to mention that once. I'll mention that several times, but that's sort of conceptually what I want you to grasp. DNA makes RNA, makes protein, because this is going to be the basis of human disease genetics. So here's a periodic table of elements. So everybody's probably seen this from your high school chemistry class, right? The periodic table of elements. Well, there are basically 5 elements: oxygen, hydrogen, carbon, phosphorus, and nitrogen. And that's all you need. That's all you need to make DNA, to make the building blocks of life, which a lot of people don't realize. It's the most simple molecule. They fall into this green category, which are not reactive nonmetals. So let's talk a little bit about the components of DNA. There are 4 DNA bases: A, G, T, and C, okay? And they're made up of different sort of components. There's this phosphate group over here. There's a sugar, it's called a deoxyribose sugar. And then there's the actual base, right? And what I'm gonna tell you is that A is always interacting with T, and G over up here on the upper right is always interacting with C. Now that DNA, which again makes up your chromosomes, it makes up all your genes, it's in a double helix. And you can imagine that's on the left-hand side here. If you're looking at a kind of you're turning a screw and you're going clockwise into the wall, that's a right-handed turn. So that's what our DNA is. And as I said, there are two strands of this DNA helix that are interacting via these bases. A always with T, G always with C. So everybody get that, right? Okay, good. So a little bit more about DNA. I said these were super simple. It's a super simple structure. Well, carbon, oxygen, and hydrogen, they're present in water and air. So really readily available, right, to make a molecule of life. 78% of Earth's atmosphere is nitrogen, right? And 1 gram per kilogram of the Earth's crust is phosphorus, right? So there you have it. I mean, these are plentiful elements, right? This is glucose, something that most people eat a lot of. I tend to limit my glucose, being a type 1.5 diabetic, but it's very similar to a sugar that's in DNA. And that's right here. This is a ribose sugar versus this glucose here. And basically, these bases, right, A with T, G with C, they're arranged in a linear order for each strand of the double helix, with A pairing with T, G pairing with C. And there are 23 pairs of chromosomes in humans, 3 billion base pairs, meaning a T, or a G, or a C. That's a base pair. And chromosomes are strings of millions of these base pairs. And along each chromosome are thousands of genes, and those genes encode proteins, okay? So what is genomic variation? So this is a really important concept, or set of concepts, before I talk about human disease genetics. So these are basically 7 individuals, and this is just some random sequence of DNA. We're only showing 1 strand of the DNA, okay? So individual 1 has a G, G, C, A, T, C, G, et cetera, all the way down part of this chromosome. Individual 2 has more or less a similar sequence of DNA, but it's slightly different, right? The slight differences in DNA between you and your neighbor, that's genetic variation, okay? So differences in the DNA from individual to individual. What makes up these differences, this genomic variation? Several things. Something called single nucleotide polymorphisms. So if you look down here, and I'm comparing all of these individuals 1 through 7 at this position 5. Can you see that? Everybody see that? Everybody has a T in that position except for 1 person. That's an A, right? So that would be considered a single nucleotide polymorphism. It's something that's a little bit different, right? And FYI, single nucleotide polymorphisms are common. We all carry like 3 million of them, and 3 to 10 million of them, and they're usually not associated with disease, okay? There are also things called indels. Look over here in individuals 5 and 6, there's a deletion of 3 bases, right? That's an indel. In means insertion, del means deletion. So we can have both insertions or deletions. That's going to be important for disease genetics. Repeat regions, you can see over here, these microsatellite sequence, it's AG, 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 right? A repeat sequence. They tend to be associated with disease as well. Anyone heard of Huntington's disease, right? They have these trinucleotides, so 3 nucleotide repeats that get amplified over time and can cause disease, right? Then we have copy number variants. These are like indels, but they're huge. They're 1,000 base pairs or greater, right? So these things can have pretty profound effects. If there's a big deletion in the genome that takes out a bunch of genes, that can potentially have a very deleterious effect. So again, a lot of these, I'm talking for the most part about common variants. They can cause disease, but most of the time they do not. I've already gone over that you differ from your neighbor by about 3 million base pairs out of the 3 billion. Okay. So let's take a closer look at a gene. So here's that nucleus again, right? We're yanking out of the nucleus. Sorry, here's the nucleus inside the cell. We're yanking out of the nucleus a chromosome. This happens to be a chromosome that's actually duplicated itself. A little bit too technical, maybe. And then if I were to pull the chromosomes, remember, they could be 6 feet if you line them one after the other. So they have to be compacted, right? And they're really condensed and compacted. If I were to stretch it out, that's what you see on the right side here. And there's all these base pairs, right? The G with the C and the A with the T. And a whole bunch of those base pairs, several thousand of them, they constitute a gene, okay? Yeah, one cell, take all the chromosomes, line them up 6 feet. All cells in your body, 67 billion miles of DNA. 67 billion miles lining up chromosome after chromosome after chromosome. So how does DNA encode life, right? So we've got DNA, and I'm saying this a second time because I really want to try to underscore these concepts. DNA is made into RNA. RNA is just a copy of the DNA. It's slightly different. So remember, there was G, A, C, T. Well, now the T is replaced with a very similar base called U. And the sugar is a little bit different in RNA. But you don't have to know that. All you need to know is RNA is very similar to DNA. It's just a copy of the DNA. And that RNA then gets transported out of the nucleus into that goo, the cytoplasm, into the goo outside of the nucleus, where it's translated into protein. And it's translated by those, remember those ribosomes I pointed out? That's the translation machinery proteins. And proteins are pretty much what do everything in your cells. Muscle proteins help you contract muscles. Enzyme proteins help you break down food. Enzymes in the heme biosynthetic pathway process the porphyrins to basically wind up with heme, right? So it's all about getting DNA into protein. DNA, make a copy of that DNA into RNA, take that RNA, bring it out to the cytoplasm, into the goo, and then have it translated into protein. You're basically reading the code on the RNA to make the protein, okay? So there's another thing that has to happen, though. So here's a kind of a schematic, a cartoon of the cell. Here's our nucleus, and here's our DNA. So I'm underscoring it a third time here. We're now going to make an RNA copy, right? An RNA transcript is what we call it. But that RNA has to get processed. So little bits of that RNA have to get chopped out to make a functional RNA. And that's that mature RNA. And that's called a messenger RNA. It's a messenger because it's going to be the messenger from the DNA to make the protein. And then that messenger RNA gets transported into the goo, the cytoplasm, where these ribosomes jump on it. This is the translation machinery to make protein. Everybody following me so far? Have I explained this simply enough? So one really important thing, because we're going to talk about this when we get into the porphyrias. So it's not just the bit that gets made into RNA that when altered somehow can cause disease, but there are sequences in the DNA that are not part of the sequence that's made into RNA. Those are parts of DNA that tell the gene when to be on or when to be off, right? And those are called regulatory sequences. So they turn on the transcription of the RNA or turn it off. So DNA makes RNA makes protein. That's the thing that you need to remember. And translation converts the mRNA to a protein. Another way to call a protein is just a different word, polypeptide. And the building blocks of polypeptides are amino acids. You've heard about that, right? Make sure you have a diet rich in amino acids, and bodybuilders, they'll take supplements that are rich in lots of amino acids. They're the building blocks of proteins. Simple as that, right? And there are 20 of them. So now we're getting a little bit more complicated, and hopefully you guys can follow me. Remember, the DNA was a bunch of G's, A's, T's, and C's. When you make it into RNA, it's a bunch of G's, A's, U's, and C's, right? And that's in a long linear line. What the translation machinery is going to do is it's going to read every 3 bases of the RNA. And that's going to be the code for what building block of protein, called an amino acid, gets inserted into the protein, okay? So this is the code right here. This is the code of life, right? So let's say in my string of bases, RNA bases, there's a CAG. Can everybody see that right here? Can everybody see where I'm pointing? That is going to encode glutamine, GLN, glutamine. So whenever the translation machinery sees CAG, it's going to add a glutamine to the protein, right? And it's going to use this code. This is exactly the code that it's always going to use, right? Now, there are a couple important things to point out about this code. One is that it's degenerate, meaning that lots of different triplet codes, they're called codons, can encode the same amino acid. So if the translation machinery sees CUU, it's going to insert a leucine, this is this amino acid here, into the protein. If it sees a CUA, it'll still insert a leucine, right? So even though this third position is different, you're still going to insert a leucine. This is going to be really important for you guys to remember. The ultimate goal I want to hand to you guys in going over this stuff is when you get your clinical testing, your genetic testing report, and you're reading, what the heck is this synonymous variant, right? What's this missense variant? What do these all mean? I'm going to tell you exactly what they mean here, okay? Okay, so degenerate code, lots of different bases can be, the bases can be different at this third position, but you get the correct amino acid put in that protein, okay? Another important thing, right? Every protein starts with an AUG. That's the first sort of part of the sort of RNA that's read. So when you're looking at the RNA, when the translation machinery is looking at the RNA, it's going to stop at the first AUG and then read in threes, and insert the appropriate amino acid into the protein. Does everybody understand that? Are there any questions? You okay? Okay. There are 3 codons called stop codons. You can see my pointer here, UAA, UAG, UGA. When the translation machinery encounters one of those, it says, okay, stop. I'm going to stop translation. I'm not going to continue. That's going to become really important when we talk about human disease genetics, okay? So now let's take this one step further, okay? So here we have a piece of RNA. Hopefully this will test whether you understand the concepts I presented. And we have this sequence ACG, UAU, GCG. Well, the translation machinery is going to look at that and say, oh, ACG, I know that I have to insert a threonine into my protein. That's that amino acid building block. Now the next triplet is UAU. That signifies a tyrosine. I'm going to put a tyrosine as the next building block in the protein. CGC, that encodes an alanine. That signifies an alanine, so I'm going to put that into the protein. Does that everybody understand that? Good. Okay. I like keep either nodding or shaking your head, Nicole, because I'm relying on you and Laura for understanding here, since you guys are the ones that have your videos on.
You got
okay.
it.
So let's take a look at what happens if we start changing some of these letters, right? So the first thing I'm going to do is we're going to look at this middle codon, this UAU, right? It normally signifies a tyrosine. But now what if you have a mutation, right? A mutation that changes this last U to a G. Uh-oh, look what happens. You've created a codon that normally encodes a tyrosine and made it a stop codon. Remember, UAG, UGA, UAA, those are all stop codons. You don't want those in the middle of a protein. So this is what's called a nonsense mutation. So even though you were supposed to have a tyrosine put in there and then move on to basically make the rest of the protein, you've actually now introduced, because there is this mutation that happened, a stop codon. So the protein stops and it can't be fully synthesized, okay? This guy here, this is another important mutation to talk about. So this is a UAC, right? It turns out that UAC also encodes tyrosine. Remember, I said that the code is degenerate. Sometimes that third position can be different, but encode the same amino acid. Great. So, okay, so this is less likely to cause disease because it's not changing the amino acid that's inserted in the protein. Everybody understand that? Right? A lot of people, when they get their DNA report, right, their genetic test, if you will, they'll find that there's a silent mutation and they'll be worried. And they'll say, look, there's a mutation here. But you have to think of mutations in their proper context. This is likely, more likely than not going to have an effect. That's not always true, but most of the time, a silent mutation will not cause a disease. Then let's look at some of these guys. I don't want to belabor the point too much, but now we've started mucking around with either the first nucleotide, the first one we're changing, right? So rather than UAU, we're getting a GAU, AAU, CAU. Every single one of those changes the amino acid. Okay, that's called a missense mutation. So a lot of mutations associated with porphyria are missense mutations, okay? And so you're basically replacing the right amino acid, here it is tyrosine, with the wrong amino acid. But guess what? Sometimes that's not the worst thing in the world. So let's look at this second set, this set of codons where we're changing the the second nucleotide. So UAU, now it's UGU or UCU or UUU, even though every single one, these are missense mutations, even though every single one is changing the amino acid, so you're changing the right one with the wrong one, some have more drastic consequences because the shapes of the amino acids are drastically different, or maybe one amino acid is acidic, one is basic. So really different properties. So if we think about that, cysteine right here and serine, these are serious changes, you know, that really are very different from tyrosine. But guess what? This phenylalanine here, that's kind of similar to tyrosine. So even though that's a missense mutation, it's changing the amino acid, it may not be as pathogenic. It may not be as deleterious. Okay? Okay, so now we're getting close. We're gonna talk about one more type of mutation. So here we have our DNA at the top that's represented. Here we have our RNA, the RNA copy of the DNA, sort of, you know, in this green bar here. And now you can see UCU right here, that encodes a serine, CAU encodes a histidine, et cetera, et cetera, et cetera. Now what we're gonna do is another type of mutation you can encounter is a deletion mutation. Now we're going to delete out this guy here. So we're effectively deleting out the U part of the CAU codon. What's going to happen is now you've shifted everything. If you can see my little camera view of me, right, you're actually basically shifting what's downstream a little bit closer, right? So you've put things out of frame. So rather than having a CAU, because you've removed the U, you go to CAG, right? CAG, that encodes a different amino acid. and it encodes a different amino acid for the rest of the proteins because you shifted the reading frame, right? So rather than having a serine, histidine, aspartate, proline, now you shifted it to serine, glutamine, isoleucine, et cetera. They really screw up the protein. So these are called frame shift mutations, and they're caused by those indels I was referring to earlier. Those can be really, really deleterious and pathogenic, okay? All right, so remember how I said I showed you that cartoon where DNA has to make an RNA, and that RNA then needs to get transported into the goo to make protein. Well, I kind of left one little step out. That RNA has to be processed. Certain bits of that RNA have to be cut out because they're not really useful for making the protein. And those bits are called introns. The bits that encode the protein are exons. And this is kind of exemplified here. If you look at this, this is a gene called tropomyosin in the human. This is the sort of RNA that's initially made from the DNA, right? And hopefully you can see, those of you that are not colorblind, if you are, the bits that are in between the numbered bits are what are called the introns. They need to be removed. And they can be removed in different ways. Depending on how they're removed, we can splice the other bits, the non-intron bits, the bits that encode protein, in whatever way we want. And literally, I'm showing you all the different ways that this big RNA transcript can be spliced to make mRNAs that then get translated in the goo, right? So this would be the isoform in striated muscle. This would be in the isoform in smooth muscle. This would be the isoform in brain, et cetera, and so forth. So everybody understand that, right? So you have this big RNA. Before you bring it out to the goo to be translated, you have to remove bits, okay? Important. So let's look at where all the mutations that cause human disease can reside. So I already told you about in these exons, right? These are the bits that are encoding the protein. You can have these point mutations, the silent mutations, right, that don't change the amino acid. You can have the missense mutations that change one amino acid to the wrong amino acid. You can have the nonsense mutations that basically create a stop in the middle of the protein. And you can have those frame shifts that are really bad, right? So, so there you go. You can also have insertions and deletions that sometimes will cause a frame shift. So that's all the stuff, right, inside the exons. But I also mentioned there may be parts of the DNA outside of the bit that's made into RNA called regulatory regions that turn the RNA on, that turn the gene on to say, make RNA. Mutations in those regions can cause disease. And there are some mutations in these types of regions that have been identified that cause porphyrias, right? So virtually every single example I'm showing you, there have been pathogenic mutations that have been identified in porphyria patients, okay? So I'm not just giving you a biology lesson, I'm giving you a porphyria lesson, right? And so in order to remember, we need to remove these introns, right? Once we make this RNA, right? These are the bits that we don't need for the protein to make the protein. And there are sequences right at the end of the exon and the beginning of the intron, right at the end of the intron, beginning of the exon, that are required to allow that to be pulled out, to be spliced out. Those are called splice site mutations. If you get a genetic testing report, they will also report those. And guess what? If those sites are mutated such that you don't remove this kind of intervening garbage, then you're going to get garbage when you try to make the protein because all the right bits aren't connected. So effectively, what I'm showing you now are all the places where you can have mutations that cause disease. And again, in every single one of these places, there's been a mutation that's been, multiple mutations been identified in porphyria patients. I won't talk about polyase site. That's just a little too complicated right now. So types of mutations that can occur, let's summarize. A missense mutation where the wrong amino acid replaces the right one. In our world, the human disease genetics world, we call these non-synonymous variants. And here's another big kind of set of scary information and terms that people will get on there when they do genetic testing. You know, variants. And when I say variants, I should also say, we used to call them mutations, right? But now we call them variants. They're the same thing, right? It's just probably a more kind of politically correct way of saying things, because mutations denote really bad things. Variants are just variants, you know, that are different. And I'll even argue that there are some disease variants that give people actually a selective advantage in life, right? A selective advantage in life. Anyway, okay, so they can be, so let's say a geneticist identifies a variant in the HMBS gene, right? He can characterize it in a number of different ways. Pathogenic, right? Pathogenic would basically mean, I don't know what this means. Pathogenic would basically mean, yeah, this is strongly associated with this disease. We have enough evidence that, yeah, you know, this really is a disease-causing mutation. Those are the easy ones. Likely pathogenic, slightly less compelling story, but probably is. Variants of unknown significance, right? A lot of people are really puzzled by this, right? These are, you know, I wish the folks that sent that provided the genetic test to you guys did a better job of explaining this stuff. And I think this is part of why I'm here right now, right? Variants of unknown significance. All it means is maybe this variant is so rare, right? We can't really say anything about it. We don't have enough information, right? So one of the things that we look at in human disease genetics is a variant, right, enriched in cases with the disease versus controls, right? That's going to provide statistical evidence that it could be pathogenic, right? If n equals 1 is one person where you found this mutation, we can't really say anything in the regard with the frequency. So frequency makes a big difference. And that's why some variants, this was in the questions that I'll answer later. I have a long list of questions that you guys sent in. Thank you for those. And Nicole has been punishing me by sending me last minute questions. One was 15 minutes, I think, beforehand, which I will answer, by the way. So there have been variants that have been considered likely pathogenic in the past that are now declassified as pathogenic, right? Because we now have more information, we have more cases, right? And controls, and we can have a better idea of frequency. So why is frequency such a big deal in human disease genetics? Because guess what, guys? Mutations that do bad things tend to be rare in the population. Why is that? Well, it kind of makes all the sense in the world. And my apologies, I need to give myself some glucose. This is my genetic disorder. My sugars are running a little high. Very standard issue here. Disease mitigation, live around the world.
We all understand that.
So yeah, so once they have enough information, you know, and look at frequencies, they can make a better ascertainment of the likely pathogenicity. Yeah, and I guess what I was saying was, you know, mutations that do bad things are going to be rare in the population. The reason is, and it makes all the sense in the world, right? People that have severe disease, they're less likely to have children, right? They may never make it to an age where they're able to have children. So, you know, this is just population genetics theory. So over evolutionary time and over generations, those variants are going to become less prevalent because they're not passed down. Okay, so that's going to be important with the porphyrias because a lot of these variants are considered rare. You know, so if you have a common mutation, you're going to, you know, that's common in the population, meaning 1% or greater, you have to really be suspect about that potentially contributing to the disease. Now, again, porphyrias are just such a beast and a challenge to the geneticist like myself because it does somewhat defy the rules. And I'll get into that a little bit later. I'll get into the weeds. Okay, so then likely benign, right? Well, those, benign are relatively easy to categorize because maybe you're going to have the same frequency in diseased individuals as controls. So if it's having some effect on disease, it should be enriched in the diseased individuals, right? The other thing is we can actually look. Remember how we talked about amino acids? So what I actually told you about is not a bunch of gobbledygook. It actually is going to become important. Remember how I said some amino acids are similar to others, right? So if you have a missense mutation where one amino acid is changed to a similar amino acid, that can be tolerated. That can actually be tolerated. And if it's not in a critical part of the protein that has a really important function. And one thing we do is when we look at variants, we put them through this software, SIFT or Polyfen2. You don't have to know the details of the software, but what it does is, one of the things it does is it looks at the evolutionary conservation. So if a chimpanzee, a zebra, a giraffe, a penguin, a human, all have the exact same amino acid at this part of the protein, if that changes, that probably is going to do something bad, because what evolution is telling us is this is a really important position. This amino acid has to be at this position. Everybody understand that, right? Versus what happens if you look at the zebra, the penguin, the monkey, the human, and there are different amino acids at that position. What it means is, it doesn't really much matter so much what amino acid is here. The protein works just fine, okay? So hopefully that gives you a little overview of how these kinds of variants are classified, right? So let's look at a synonymous variant. Remember, that doesn't change the amino acid because it's at that third position of the codon, right? And because you could have different bases at that third position and you get the same amino acid. These are very unlikely to cause disease. So if you see a silent mutation, a synonymous variant, it's less likely to cause disease. Don't get me wrong, there are cases where they can cause disease. I won't go into the complexities of how, but they're less likely at least, right? And that's sort of another thing to say about human disease genetics. We're talking in the realm of likelihood. That's the problem. I think a lot of people want, and understandably so, definitive answers. And unfortunately, in human disease genetics, and especially with the porphyrias, it's really nebulous, and there are a lot of nuances. And I'll hopefully bring a lot of these up to you, all of these up to you today. So nonsense mutation, premature stop codon created, protein can't be translated anymore. You don't make all the protein. That's bad, right? Splice site mutation, where we have to take out the bits that we don't need of that long RNA, that inhibits splicing. You get translation normal up to that part that's not spliced out, and then a bunch of gobbledygook. That's also bad. That's also bad. Frame shift mutation, we talked about that. I took that one base pair out. It shifted the frame, and then there's gobbledygook from there on out. That's bad. And that's caused by indels. I won't go into this, probably too much information. Let me see how the time is. Wow. Okay, we're cranking up the time here. Hey, Nicole, how important is it to stop at 5?
It's up to you, however much time you have. So
okay,
we're here to listen to whatever however much time you can give us.
I understand, guys, if folks have to leave, but fortunately this will be recorded. I'm going to get into the porphyrias. I promise. I'm going to get into a lot of great detail on the porphyrias. But as you can see, I think you need to know some of this basic stuff, right? And then I mentioned about the regulatory mutations. Okay, so this is what we do in human disease genetics. We look at pedigrees. If you look in the kind of lower right here, males are squares, females are round. Affected individuals are darkened squares or circles. And these are the pedigrees. So this would be a man and a woman having 3 kids. They're sort of arranged in birth order. Had a son that's unaffected, a son that's affected, and a daughter that's unaffected, right? So that's how you read these kinds of things. And where this arrow is, that's called the propositive. It's also called the proband. And that's sort of just a term we use in human disease genetics. When we talk about sort of families and the individual that sort of that family that got us into studying this disorder. So Mendelian disorders. Okay, when we talk usually about human disease genetics and rare genetics, rare diseases, right? Porphyria is a rare disease. We talk about a Mendelian disorder. And Mendelian disorders, they follow the rules of Gregor Mendel, this Austrian monk that did all this work on pea plants, right? So what I'm showing you here is an inheritance pattern. It's a disease that's inherited in an autosomal dominant fashion. Now, you guys have heard, if you guys have AIP, right? Autosomal dominant, right? So this would be relevant to, in some ways, to your disease, where affected individuals have a 50% chance of giving it to their children. So 2 out of the 3 children have it here. And those children that are affected, 2 out of the 6 here are affected, or 2 out of the 6 here are affected, 2 out of 3 here are affected. The problem with porphyrias is that the penetrance can be low, it can be reduced. In this case I'm showing you here, the penetrance is high. Man, you get the mutation, you're gonna get the disease, right? That's not necessarily true for the porphyrias, because what I'm gonna show you, especially for the ones that are inherited in an autosomal dominant fashion, what happens is one of the copies of the enzyme, the gene encoding the enzyme of interest, is mutant. The other is completely fine. So the worst you can do is have 50% gene function. 50% gene function, right? Boy, that drives geneticists and families crazy, because what that means is it's kind of this teeter-totter area, right? And this is an area of genetics that I have quite a lot of expertise on, because this is, in fact, how we identified, we published a big study last year in the American Journal of Human Genetics. This is how we identified a bunch of novel cleft lip and palate genes by looking at these genes that one copy was removed and the other copy was fine. So you're on this teeter-totter level. And these are genes that are called haploinsufficiency genes, right? So having one copy is not enough to prevent you from potentially getting a disease. Most genes in your body, most genes on your chromosomes, you are absolutely fine having one good copy and one bad copy, right? In fact, I would say probably virtually all of us on this Zoom have disease genes, other disease genes, forget about porphyria, forget about diabetes, that are never going to manifest themselves because you have another good copy of that gene, right? So with the porphyrias, you have one good copy, and then you have, so this will be the good copy. You see my video here. This will be the bad copy. The bad copy, depending on the pathogenicity of the mutation, it can reduce it 50%, 70%, 80%, even 100%. So there's this incredible variability. So the bottom line is, even though you can have AIP, right? Even though you can have AIP, depending on the pathogenicity of that mutation, that reduces the amount of function of the mutant allele. That makes a big difference in terms of whether your other family members and whether you will present with a more severe disease, right? The other thing that's really, really important is genetic background. Why do not, let's put that aside. That's concept number 1. Concept number 2 is genetic background. Everybody's different genetically, even in the same family. I won't go into basic genetics that proves that, but everybody's different. And so you have basically this genetic background effect. In some people, your disease is more manifest, right? And then thirdly, environmental situations, environmental influences. As you guys know, right, there are certain things you don't want to do if you have porphyria, right? You don't want to drink alcohol generally in excess, it's certain drugs you don't want to take, right? So there we go. So I'm just going to blow through this. So let me talk just a wee bit about, because I mentioned this before, environmental influences. You guys have probably heard about these hemoglobinopathies, right? So in particular, the sickle cell disease, otherwise known, some people call it sickle cell anemia. There's a hemoglobin variant that carries red blood cells that are deformed into a sickle shape when oxygenated. And this shortens the lifespan of cells, can cause blockages of blood flow, extreme pain, et cetera, and so forth, right? And of course, this connects immediately to the porphyrias because the heme group is part of hemoglobin, right? That's what carries oxygen around your body. Beta thalassemias are somewhat related. Individuals with beta thalassemia present with severe anemia. Variants reduce the production of hemoglobin. Well, guess what? Both these and other disorders of the red blood cells are by far the most common genetic diseases in humans due to the strong selective pressure for resistance to malaria. So people that carry these mutations are actually less susceptible to severe malarial disease. There are some hypotheses as to why that's the case. I'm not going to go into that now, but suffice it to say that, what's the disease mutation here, right? The disease mutation is actually giving these individuals a better chance of survival. So I love that example. So don't ever think, right, that just because you have a disease variant, it's abnormal, right? All right, and so this is an important kind of graph to sort of go over because it really underscores disease genetics in a nutshell. So we call it the common disease, common variant, rare disease, rare variant hypothesis. And it's as simple as this. Common diseases like most types of diabetes and high blood pressure, these sorts of things, they're caused by common variants. So they're floating around out there. But you might ask, well, why aren't they selected against? Because any one of these variants that's maybe slightly compromising a gene function, it's not doing anything. It's not causing anything bad in a person that inherits it. If you go to the next gene that might be involved, that's not doing anything harmful. It's only until a person inherits a whole bunch of different variants in different genes that they get the disease. So there's no real selective pressure to get rid of them. So common variants, so mutations that are common in the population, are what cause common disease in general, okay? It's those rare alleles, those rare mutations that cause the really nasty stuff. I already explained to you why they get weeded out. So some of the porphyria alleles, the porphyria mutations, are in that area, right? They do really bad things and they're rare. But the problem is, right? What about these guys that kind of only slightly reduce the enzyme function and you're maybe on a teeter-totter, you might present with the disease, and if you do, it's not severe. Well, those might be variants. If you look at the chart now, they're kind of not rare, but maybe lower frequency with sort of intermediate effects, and maybe kind of edging into common variants. There might be a couple common variants that might edge you a little bit toward the disease. So important information for you guys, because that is oftentimes an explanation for why a certain family, even though they have a variant and it may be pathogenic, you may have very mild disease, okay? So now we get to King George III, right? I think some people were looking forward to this part. It's very basic. It's like 2 slides. Reigned from 1738 to 1820, presided over a turbulent period of British history. That was when American independence happened, French Revolution, Irish Rebellion, whole bunch of cool stuff. So King George III suffered from series of bouts of madness that were incapacitating. During each attack, attending physicians would note a rapid pulse, fever, yellow or bloodshot eyes, abdominal cramp-like pain, constipation, cramps in the legs, skin blistering on his arms, port wine colored urine, pretty kind of, ding, 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 right? And rambling speech degenerating into obscenities and hallucinations led to the moniker, the Mad King George. Various treatments were tried, including bloodletting with leeches. I don't think any of you guys have leeches in your cupboard. I hope not. In 1810, after descending into permanent derangement, was deemed unfit to rule and died in custodial care in 1820. So let's talk about the genetics, diagnosis, and clinical features. So I don't know. So this is what I found in the report that I found. They diagnosed him with porphyria variegata or variegate porphyria. First of all, I think they got it wrong for obvious reasons, right? The subcutaneous lesions, right? But we can talk about that later. What's interesting is these types of symptoms could be attributed to his relatives, Mary Queen of Scots and Lady Stuart, and his son, King James I, thus suggesting a genetic component. It's a rare disorder of autosomal dominant inheritance with low penetrance, okay? And so I think a big discussion in the porphyria community as well, there are so few individuals in this family amongst these relatives that have clear signs of porphyria. It can't be genetic, right? Or it can't be porphyria. Well, if you start to really understand the genetics of porphyria, this makes complete sense, right? That some individuals would inherit the mutation and have porphyria, the clinical features of porphyria, and others may not, right? This very kind of low penetrance kind of phenomenon. For variegate porphyria, the gene is PPOX, encodes an enzyme needed for the production of heme for use in hemoglobin. This is just taken right from my course. Clinical features of individuals, not just variegate porphyria, but in general porphyria, constipation, abdominal pain, tachycardia, that's quickening of the heart, hypertension, high blood pressure, muscle cramps, photosensitivity. And by the way, that's, the avoidance of light. And there were vampire associations ascribed to individuals that had porphyria, skin blistering and rashes, confusion, fever, and rarely psychosis. And of course, we all now know it's due to buildup of intermediates in this heme pathway, right? Porphyrins or porphyrin precursors, right? Because technically they're two different things. And I threw this in because this is kind of a fun little aside. There's a disorder called hypohydrotic ectodermal dysplasia. It's an X-linked recessive disorder. It's in this EDA gene, and individuals present with, abnormalities in ectodermal structures. That just means sort of outside of the body. So hair, teeth, skin. There's dark around the eyes, prominent forehead, et cetera, et cetera, et cetera. And here's an individual with a cutaneous form of porphyria, right? These are individuals with HED, right? And you can see they have very pronounced canines. And so both of these disorders, both porphyria and HED, really fed this ridiculousness of the vampire disease, right? This is the Cullen family in England. And you can see mom is unaffected. The two kids are affected. Those of you who are kind of nerds, genetic nerds, this is an X-linked recessive. And so she has one good X gene, the EDA gene, and one bad gene. That's why she's okay. This is not a case of haploinsufficiency, but when she gave her, she has to give an X chromosome to her sons. She clearly gave the defective one to the sons, and they're affected because they only have one X because they got the Y from dad, right? Okay, so now we're able to get to the porphyrias and its many forms. See how we're doing here. Okay, actually, we're not too bad. We're only a few minutes over time. So porphyrias are inborn errors of heme biosynthesis, right? Remember, heme is a structure that basically houses an iron molecule, and that gets incorporated into a number of different protein complexes, such as hemoglobin, right? And as well as enzymes like cytochrome P450. There are some oxidative stress enzymes. You build up antioxidants in your cells, and you need to get rid of those. So heme is associated with some of those key enzymes. And I have a pet theory that we can talk about at some other Zoom, that I wonder whether disruptions in heme biosynthesis are affecting your ability to basically mitigate oxidative stress, right? And whether that's contributing to the clinical features. But this is at this point only sheer conjecture, right? So we all know, right, 8 major disorders due to either loss of function or gain of function, e.g. XLP, mutations in 8 of the 9 heme biosynthesis genes, which all encode heme pathway enzymes. I should add that PCT, the most common cutaneous porphyria, it can be sporadic and it has nothing to do with enzymes. Mutations. And it has everything to do with an inhibitor that gets made in the cells and that can basically inhibit UroD, right? So there are 3 major categories of the porphyrias: the acute hepatic porphyrias. I'll break those down in a second. Hepatic cutaneous porphyria, porphyria cutanea tarda, or PCT. I will refer to them by their abbreviations, PCT, for example. The cutaneous erythropoietic porphyrias. And a lot of people don't really understand what the difference between the hepatics and the erythroid porphyrias are. I will explain that. Hepatic porphyrias are due to a buildup of heme precursors in the liver, right? Whereas erythropoietic porphyrias are due to buildup of heme precursors in the red blood cells and their precursors. That's called the erythron, E-R-Y-T-H-R-O-N. So, and there are some porphyrias where there are defects in both hepatic and erythropoietic, right? The acute hepatic porphyrias include the acute intermittent porphyria, AIP, hereditary coproporphyria, HCP, variegate porphyria, VP, and I don't like pronouncing this, but 5-aminolevulinic acid dehydratase deficient porphyria, which will abbreviate ADP, right? So those are the acute hepatic porphyrias. The cutaneous erythropoietic porphyrias are the erythropoietic protoporphyria, EPP, congenital erythropoietic porphyria, CEP, and X-linked protoporphyria, XLP. There have been a ton of mutations, right, that have been found in these heme biosynthesis genes causing porphyria, as have their kind of modes of inheritance. So remember I said that there are 2 places where heme needs to be made, and they're here, right? There are the hepatocytes. Those hepatocytes, AKA liver cells, right? And there is the erythron that I mentioned. Those are in the bone marrow, the red blood cells and their precursors, right? And they basically make use of most of the same enzymes. The one significant difference is this first enzyme. If you can see my pointer, in hepatocytes, it's ALAS1, and in the erythron, it's ALAS2. And so you can imagine, right, that mutations that affect, ALAS1 are going to cause porphyrias potentially in hepatocytes, whereas mutations that affect ALAS2 could potentially cause porphyrias in the erythron. And also how these particular genes are regulated, right? They have, remember those regulatory elements I told you about? I told you needed to follow me because it's going to come back to haunt you. Those regulatory elements that control expression, well, there are regulatory elements that control expression of these enzymes in the liver, and there are regulatory enzymes that control, regulatory regions that control expression in the erythron. So mutations in these different areas can affect regulation in one versus the other, okay? So hopefully everybody gets that, right? Okay, and I love this slide because I kind of annotated it. It's from this Dickey et al. Annual Reviews in Medicine. I've put here the compounds that build up in the various porphyrias, right? immunolevulinic acid right here, otherwise known as ALA, right? Porphobilinogen or PBG. You guys are probably very familiar with these two. And of course, these out here are going to be the genes, the ALAS genes, the ALAD, HMBS, causing, of course, the AIPs, the Uros, Uro-D, CPOX, PPOX, and FEC or FECH. And these would be the porphyrias, right, that are in this sort of inner red, XLP, ADP, AIP, et cetera, and so forth, right? And so this next slide is not particularly great. It leaves out a couple of porphyrias, but the reason I left this in, I sort of annotated it for you guys to denote which ones are actually, which ones these represent. Notice that in the lower right, this is a liver over here, and this is kind of the bone marrow, right? And you can see, for example, right, that EPP, that's a bone marrow sort of erythroid specific porphyria versus variegate porphyria, which is a liver specific type, right? Whereas PCT, right, cutaneous tarda, that's defects in both, right? And so this is going to be reflected in where you have your clinical presentations, right? Because it's the blood cells that are circulating, the red blood cells, they'll die and they'll release these porphyrin precursors, and then when kind of illuminated by light, it will cause pain, right? So those will tend to be associated with the cutaneous, right? Features, clinical features. So acute hepatic porphyrias are due to loss of function mutations that decrease the activity of one of the heme biosynthetic enzymes. 3, AIP, HCP, and VP show autosomal dominant inheritance patterns. And I told you what that means. One good copy, one copy where there's a mutation that reduces the function of that gene, that particular copy of that gene, depending on the pathogenicity of the mutation, to variable degrees, right? ADP shows an autosomal recessive inheritance pattern. What does that mean? That means that both copies are actually mutated, right? That doesn't mean that they kill the gene function, no, right? What it means is one copy is dropping it down a certain amount, another copy is dropping it down a certain amount. And in that case, with ADP, you can actually get to a place where you're potentially below 50%. With the AIP, HCP, and VP, you can only get down to 50%, right? Because remember, there's only one copy of the gene is mutated. The other that's making 50%, right, of the enzyme is just fine, okay? So important, with this type of autosomal dominant inheritance, one gene copy is mutated, whereas a second gene copy is not. Therefore, there is a reduction, but not absence of enzyme production. Some porphyrias may have very low penetrance and can be difficult to diagnose. This is one of the reasons why. CEP and EPP show autosomal recessive inheritance patterns. So again, mutations in both copies of the gene, right? Whereas XLP is an X-linked inheritance pattern, and due to gain of function mutations, right? So you're actually adding to the function of the protein. I don't really want to go too much into explaining gain of function because that's going to bring me down a rabbit hole. But it's a slightly different genetic phenomenon. PCT can be acquired, sporadic form, or show an autosomal dominant inheritance pattern. So just like the AHPs, right? So one good copy, one copy that's mutant. But what's really interesting about PCT, if you have the acquired sporadic form, you don't have a mutation, right? You don't necessarily have a mutation in any of your heme biosynthetic pathway genes. It's just that there's an inhibitor, right, of Uro-D that gets synthesized and it inhibits it. So it's kind of a secondary effect, but it can cause porphyria. The most common acute porphyria is AIP, and it's caused by mutations in HMBS, right? I know a lot of you, maybe the majority of you, have this particular form, certainly if you have an acute porphyria, and the most common cutaneous porphyria is PCT. I've already said this, but I'm going to emphasize it again. Most common, when I say common porphyrias, doesn't mean common, right? All porphyrias are rare disorders, and thus most variants, mutations, associated with them will be rare in the population. But I've already explained to you that kind of low penetrance variants can basically rise in frequency or can stay reasonably elevated in frequency because they're not doing horrible things. In many people, they can maybe be tolerated, right? But then there's a phenomenon of what we call a population bottleneck or a founder effect. You guys may have heard about this. So there's a variant that is increased in Norway and Sweden. It's in HMBS, right? So it would be AIP, right? Why is that? Why is that? Because we already said that, you know, it should likely kind of shift more toward the rare side. It's likely going to be doing something pretty bad. Well, what happened was when Norway and Sweden were founded, as we migrated out of Africa, and we all did, to populate the globe, when the last group, when they settled into Norway and Sweden, there was what's called a founder effect. In fact, one of the small population that came to Norway had a mutation in HMBS. And so now that variant gets propagated and it starts to look like a common variant, right? It's getting higher into frequency. Importantly, if we look at that variant across all populations, it's not common. So remember how I said it's really important, really, really, really important to really consider all of these things and to have power to look at big populations when we want to understand whether a variant is associated with disease, okay? So clinical features of porphyrias, they're due to buildup of intermediates in the heme biosynthetic pathway. They're referred to as porphyrins or porphyrin precursors. They become porphyrins when they have this sort of ring-like structure, right? The precursors would be before they form that ring-like structure. The clinical features are variable depending on expressivity of the disease in a particular individual and on the type of porphyria. So what we will often say in some cases where there's a variant in a family where some people express the disease, some people do not, and those that do express it to variable extents, we call that incomplete penetrance with variable expressivity. And again, that can be a nightmare for the geneticist or the clinician, right? When you're trying to see, you know, real obvious disease transmission, sort of genetic transmission of the disease. Because look, all these people have variants, the same variant, but some don't seem to have the disease. And the ones that do, it's variable, right? When we identified that key renal agenesis gene, where that was autosomal dominant as well. Some individuals that inherited that, 2 perfectly normal kidneys. Some that inherited the same mutation in the same family, only 1 kidney. Some that inherited that, unfortunately, were stillborn, no kidneys, right? And so that's another example of incomplete penetrance with variable expressivity. It can, you know, make clinicians and some geneticists sort of lose sleep, right? Okay. And then you guys know, obviously better than I, acute neurovisceral attacks, brain fog, insomnia, fatigue, severe abdominal pain, tachycardia, remember racing heart rate, hypertension, motor weakness, seizures. For cutaneous versions, rashes, skin blistering, and pain after sunlight exposure. And that's, again, due to the sunlight actually sort of activating the porphyrins, right? Environmental influences can play a significant role in the clinical features. For AHP, attacks can be triggered by dieting or fasting, various hormonal fluctuations, especially in women, and where, by the way, you probably know there's a higher incidence of porphyria, alcohol consumption, various infections, smoking. And we've seen this too for our cleft lip and or palate study. Smoking is a great contributor, right? So this is maybe a little bit more information than you guys may may want to know, but I think some of you might be interested in this because, a lot of people are wondering, well, why do these attacks get so bad? Why are these environmental influences making the attacks so bad? So basically, remember this ALAS1 gene, right? ALAS1 gene, it's really starting the whole pathway, right? And if the cell is sensing that there's not enough heme being made, it's going to activate that gene, right? The cell is sensing it, right? So a number of these factors lead to inappropriate induction of this ALAS gene, right? We don't necessarily know how, but we're just ramping up this gene. That's allowing for the respective enzyme deficiency to become rate limiting, leading to further reductions in heme production. What do I mean by that? You're now taking a broken pathway and you're ramping it up. You're actually activating it even more, which mucks up the motor works, if you will. And so now the cell, and that leads to even less heme production. Now the cell is like, oh my God, I need more ALAS1 to activate the pathway to a higher extent. So now I upregulate ALAS1 further. Wow. Then you're in this major kind of vicious circle, leading to an even greater buildup of porphyrins or porphyrin precursors. So that's the explanation, likely, why a lot of these environmental influences contribute and can make attacks that much more severe. Okay, so why are they difficult to diagnose? Porphyria may manifest with broad and unspecific clinical symptoms that can mimic other disorders, extreme variability of symptoms due to precursor buildup in the erythron, the liver, or both, right? Most patients have reduced function of relevant genes, right? So that's not completely off, right? So you can have this variable penetrance. Some people present with the disease, some people don't, some people present with variable severities, even in the same person, right? In some attacks are going to be more severe than others. Really key, performing the proper tests at the proper times, right? So biochemical tests like the spot urine test, right? There are also tests that can be done. And I'm not an expert on knowing the availability of these tests. Nicole might have a better understanding of these. Somebody on this call might. But there are various tests that can be done. You can do these chromatographic tests where you analyze very precisely urine or blood or even feces. And guess what? And I'll tell you in a little bit, this makes a difference in terms of what porphyria or porphyrin or porphyrin precursors you can detect, okay? Proper genetic testing, right? Were the proper genes interrogated, right, when you had your genetic test? Where did they look, right? Most people are going to look for those exons, look for the missense mutations in the indels and these splice site mutations, right? The problem is we don't really know very well where to look in the regulatory DNA because that's sort of a big black box for those of us that are very active in trying to understand that part of the DNA. So it's possible, it's absolutely possible that some mutations are in those regions, right? We know some mutations have been identified in regulatory DNA that are associated with porphyria. What I'm saying is there's a pretty vast sort of space in that realm that's relatively unexplored, okay? Common versus rare variants, right? Can you really now truly rule out, what if it's an intermediate frequency variant, right? Because of everything I told you, it muddies the waters, right? VUSs, could they be pathogenic? Possibly, I would like to see if someone says this is a VUS, I would like to see on a genetic report, okay, did you run it through SIFT or Condell, for example? Is this changing a very conserved amino acid to a non-conserved amino acid, right? When I see some of these genetic testing reports, and the descriptions and ClinVar or something, I'm left unsatisfied, right? I'm left unsatisfied. Then there are new genes, right? Are there new genes? Well, we know that, remember that elase enzyme? There's a gene that's required to basically break down the enzyme when it's done functioning. Well, mutations in that gene can cause porphyria-like symptoms, right? Because you're not getting rid of that enzyme. It's done its thing. It needs to be degraded. There's a gene called GATA1, which is responsible for basically erythropoiesis, making those red blood cells. Mutations in GATA1 have been associated with porphyria features, right? So that's one of the questions I'll address in the Q&A. So yes, there are other genes and probably more down the line that could be involved. Have some of the variants been misclassified? Yes, right? We already know that, right? It's not until we get enough information from enough individuals, cases, meaning patients versus controls, that we can assign frequencies to these kinds of things. Environmental factors play a significant role, right? So you know, yeah, and the acuteness of the attacks and being able to kind of get the test as quickly as you can after the onset of an acute attack. And then there is this possibility, right? You may not have porphyria, even if it seems you do, right? We can only do so many things, you know, from the biochemical to the chromatographic tests. There are some enzyme tests, you know, the spot urine tests, the genetic tests. But if you don't have a buildup of precursors and you haven't found any variants, you really need to think about the possibility that you may not have porphyria. So with that, these are the some of the references I used, and I will start answering questions now. So I'm going to stop my share. Okay, I have a few pages of questions here. This may take a little while. You can blame Nicole. Okay, so first one. Some researchers have suggested that Lyme could cause genetic mutations. Is there any truth to that? I would say probably not. So Lyme is caused by bacteria called spirochetes, and most of the clinical features of Lyme are due to inflammatory processes against the spirochetes. Okay. All right. Diagnosis DX with PCT plus 4 EuroD. What does this mean? And this is a perfect example of a patient not being properly informed on what this means, right? Should she have other concerns? Is this the only form of porphyria she could have? Will her kids inherit it? So diagnosis of porphyria cutanea tarda, right? DX, that's what that means. And plus for EuroD, I'm presuming that means they found variants, pathogenic variants in EuroD. So yeah, it probably is PCT, right? And let me see, one thing I wanted to mention, I'm not going to put the Zoom on. So if she has EuroD mutation, so she has sort of the genetic version, right, versus the sporadic spontaneous version, acquired sporadic form. So 50% chance, right, that she's going to pass the mutation to her kid. And so there you go. But we've talked a lot about incomplete penetrance, variable expressivity. So anybody who has this sort of autosomal dominant version, that doesn't necessarily guarantee that your child is going to have severe porphyria. Okay. HFE gene mutations in acute porphyria, how many possible genes are still unknown? I talked about a couple genes, there may be more. HFE, by the way, is a hemochromatosis gene, right? And it's associated with hereditary hemochromatosis, and that causes severe liver disease, and it's due to excessive absorption of iron, right? Dietary iron. And it can basically cause porphyria symptoms, right? And so oftentimes it's misdiagnosed as PCT, but really the underlying disease is this hereditary hemochromatosis. That still means you're going to have this potential buildup of porphyrins or porphyrin precursors, and you have to deal with these symptoms. It's just that in that case, you're not going to find any variants in, the heme biosynthesis genes, right? So this could be, a definite gene to look at as one of these other genes. Let's see. The harmful effects of rare disease are what make the news. If there were no benefit to low penetrant siblings, many conditions would die out. Please provide the positive aspects referring to the spectrum of genetic effects. Positive aspects: incomplete penetrance with variable expressivity. This is a good thing because, in general, because you're not decreasing the maximum for these autosomal dominant versions is you're going to still have 50% enzyme function, right? And you can try to mitigate the attacks by controlling your environment, right? Genetic background, of course, is going to play a role. Nothing we can do about that. Severity of the mutation does make a difference, and I mentioned environmental factors. Okay, please explain genetically diagnosing AIP when the gene sequencing is unavailable or has not yet been discovered. Maybe asking about variants of unknown significance. Possibly the gene is only for that family or parent and offspring, mother and daughter both having the disease. So yeah, I mean, your first laboratory test would probably be the spot urine, right? You want to get it done right away after an acute attack. You're going to look for elevated ALA and PBG, right? You can sort of, and again, I don't know how widely available or how expensive these are, but you go the chromatographic route where you could look at urine/blood/feces, depending on the type of porphyria. In terms of genetics, the one thing, remember how I mentioned about de novo mutations in neither parent and a kid could have it? Oh, well, why do I have it and none of my parents have it, right? I don't get it. It could be a de novo mutation, especially for an autosomal dominant. All you need to do is hit one version of a gene, right? You have two copies of every gene, one from mom and one from dad. So it's, in theory, absolutely possible. Neither of your parents have the porphyria. You have it because you received a de novo mutation in one of the biosynthetic enzyme. All right. So this is a long one. PPOX is associated with variegate porphyria. I have the PPOX mutation. According to several different genetic logs, this is a benign mutation. Blah, 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 blah. Therefore, would this variant be subordinated for diagnostic purposes in the presence of classic neurological symptoms, but without a current demonstration in elevation of PBG or ALA? In my current diagnostic evaluation, we have not been able to identify an elevation in either, predominantly due to a lack of timely testing on behalf of medical staff. Super important, right? Timely testing. This won't be a particularly satisfying answer, I'm afraid. Yeah, so it's definitely true that some variants originally thought to contribute to porphyria have been removed from that disease list based on the fact that they're now been found to be too common to cause a rare disease. So one possibility that, and I've seen this in a couple scenarios where there's a variant that was previously ascribed to be a pathogenic variant, but some individuals, they're actually homozygous for that variant. Even though supposedly only one mutation should cause disease, but now people are homozygous and they seem fine. and this gets into really complicated disease genetics, but these common variants, there may be another variant that in the same block of DNA that gets inherited together. It arose on that piece of DNA. We call that a haploblock. And so it's kind of acting as a flag. So in one population, you see this, but that's not the causative mutation. It's somewhere else potentially in the gene, right? Whereas some populations, they didn't have that deleterious mutation elsewhere. So this, I dropped all my sheets here. So this, it gets to really complicated human disease genetics. And so that's why I think it's very hard to really satisfactorily answer that question. I'll also say that you can take, for any variant, if you're curious about it, partner up with a geneticist or someone who knows a little bit about how to look for, conservation of the protein structure, and you have them maybe do a little bit of an analysis. Remember how I said, if it's a variant of unknown significance, boy, if a lot of the amino acids that are at that position in the protein are invariant, gorilla, kangaroo, emu, duck, pig, human, right? And now all of a sudden you have this amino acid change, right? Even if it is a variant of unknown significance, that's tipping you more in the pathogenic direction, okay? Okay, good. Okay, so let's see. HFE, genetic mutation, has commonly been associated with several diseases, such as hemochromatosis, Alzheimer's, PCT, VP. Of most interest is Alzheimer's disease. Are those with porphyrias more likely to have early onset or faster progressing Alzheimer's? This is a great question, right? We talk about what are called comorbidities in the disease world. So if you're if you're an autism spectrum disorder individual, you have a 30% chance of having seizures, right? They're linked, and the pathology is likely linked. The genetics is linked, right? I don't think there's any obvious connection between the porphyrias sort of genetically and Alzheimer's. These are very different disease processes. But, right, Alzheimer's affects, brain processing, and has cognitive decline over time. Well, any disease that has sort of visceral features like porphyrias can certainly exacerbate, I would say. And I'll leave it at that. Right? So no obvious connections, but sure. I mean, you have two different disease processes that are affecting a neurological process, and so it could make it worse. Let's see. In regards to all testing methods for porphyrias, where would you rank the accuracy and reliability of genetic testing compared to the others? It really is, the gold standard. If you can find a pathogenic mutation, right? If you can find a pathogenic variant in a heme biosynthetic pathway gene, man, you've found that smoking gun, right? Of course, I've brought up, the issue of have you sequenced all the right genes in all the right places, blah, blah, blah. Now, the biochemical and chromatographic tests now that are being done are really getting better and better. And looking at some of these chromatographic tests, I mean, you can see these peaks of all the porphyrin and porphyrin precursors, in blood or fecal or urine, samples. It's really quite outstanding. So I think we're getting a lot more sophisticated in identifying these. Okay, let's see. Okay, patient has variegate porphyria as well as other family members. Should those who have no outward symptoms of the disease be concerned about developing an acute attack at some time in their lives? Should they be concerned about any prescribed drugs they may take? Any other concerns for them? Well, again, incomplete penetrance, variable expressivity, that seems to be the theme of the day. So The good news is people can inherit a disease mutation, a mutation causing porphyria, and have a less severe clinical presentation than, say, a parent. But Nicole also contributed some information to this, and I 100% concur, but I wanted to throw this out. This is a contribution from Nicole here. Okay, people diagnosed with acute porphyria like VP who do not have symptoms should educate themselves about the triggers for acute porphyrias to avoid bringing on an AHP, acute hepatic porphyria attack. Some triggers are alcohol, smoking, unsafe drugs, illegal drugs, extreme dieting, low carb diets, and fasting, hormone fluctuations, especially in females 2 weeks before their cycle starts, stress on the body from infections, surgery, or illness, and psychological and emotional strength. Right? So yes, 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 yeah. You know, one of the drags of living with a genetic disorder, everyone virtually on this call knows what I'm talking about. I have one. Unfortunately, one of the things that comes with it is just a tremendous amount of stress, right? And anxiety, depression, the whole 9 yards, because we're fighting, you know, for our wellness every single day. You know, over the course of this, I've been checking my glucose levels, you know, to make sure they were too high. I'm making sure they're not bottoming out so I don't go into a hypoglycemic attack, right? But I'm preaching to the choir here, right? So, you know, to try to mitigate or limit the stressors can be a really challenging thing, right? So I really get that, but trying as best you can to mitigate those environmental influences is great. Okay, so here, patient has AIP and is a biologist. Oh, this is fun. How does a mutant gene produce normal proteins, sometimes in defective proteins, at other times, not what triggers the change, but a general idea of the mechanism, if that's possible in molecular biology? Well, remember, for the autosomal dominant versions, there's no sort of abnormal protein per se. I mean, it's more, there's an abnormal protein, but it's just reducing the function. 50% of the protein is completely normal function, right? You're effectively just reducing the effectiveness of that second, right? So it's different if you're talking about some dominant gain of function mutation. That's a whole nother story. And generally speaking, the gain of function mutations are few and far between in the porphyrias, although there are some that I mentioned, a couple that I mentioned. So hopefully that answers that question. If a DNA result is inconclusive, could I have porphyria? Is there any other way to get conclusive answers? I have had the blood and DNA tests. Okay. Yeah, so what did I write here? You know, I talked about it, some additional genes that could be looked at. You know, we can only do what we can do based on the information we have, right? Looking at the bits and parts of the genes that we think are most likely containing variants. Are we missing some bits and parts? Probably. But I think really a combination between, you know, the genetic testing and some of these really accurate tests to look for the porphyrins and porphyrin precursors, that really should get you where you need to go. Okay. So this is something that I found super, super interesting that you guys, I think, might be really interested in. So remember, I listed all the precursors and I abbreviated them, like, you know, proto and copro. You guys know what I'm talking about, right? ALA, PBG, URO, right? Well, here's what's interesting. So if you do a chromatographic analysis, it turns out that some precursors, such as copro and proto, they're hydrophobic. What that means is they don't like water. So they're going to be less represented in a urine sample, but they're going to be better represented in a fecal sample. And there's a question I'm going to answer in a couple of minutes that deals exactly with just this. Okay, let's see. Due to lack of time, I'm going to skip a couple questions and we can pick it up maybe at some other point in time. here's a question I really love. Is there a genetic reason family members can have much different or much more severe than others? I really have kind of, talked about this with the incomplete penetrance and variable expressivity. That really, I think, nails it. Okay, here's an important question. So how can a genetic test be positive for EPP, right? So variants in EPP were identified pathogenic, but clinically patient tests positive for HCP. How do you know with certainty that you are diagnosed with the right form of porphyria? So my question for that person that asked it was, does the patient have a buildup of this protoporphyrin 9, right? If he or she does, that's absolutely consistent with, EPP. If not, did the test look for, or was it able to identify this precursor, right? So I found a paper where, and remember, if it's EPP, one needs to look at the erythrocytes, the plasma, or the fecal samples, not the urine, right? To see if the precursor is elevated, right? In a paper from an Italian group, this is one of the references I put, to Piero et al., urine from a patient with EPP was analyzed, and the last precursor in the chain that was able to be identified, and this was using chromatography, was coproporphyrin 3, right? Was not able to find the protoporphyrin 9. However, when they did a fecal chromatographic analysis of that patient, they found it. So it has everything to do with sort of the fact that, remember I said hydrophobic versus hydrophilic. So these protoporphyrins are better represented in the feces than in the urine, right? So just kind of knowing these subtle, differences, I think is really, really, really important when you talk about this. Okay, maybe I guess I've already gone way over time, but maybe I'll stop there and I'll hand it off to Nicole.
Well, thank you so much, Dr. Manak. You've spent way more time than you originally had planned. So thank you so much. I know everybody appreciates all your time. This has been, I've learned so much. It's just, thank you so much for sharing everything and answering all these questions. And so everyone, have fun with Rare Disease Week. Show your rare, post on social media, create awareness. And if you have any other questions, just send them to us. We'll do our best. But Dr. Manak, you've been absolutely amazing, and I'm speaking for everybody here that we are just so grateful, especially on a Sunday. So thank you so much.
Yeah, it's my pleasure, and I'm so sorry I didn't get to kind of answer the questions in the chat, because I was noticing there are just a bunch of really terrific questions. I would love to answer every single one of them. So maybe we can I can put together some answers for some of them and work with Nicole to get you guys some of these answers.
All right. Well, thank you again. Have a great week, everyone. Dr. Manak, we appreciate it so much. You're amazing. Thanks.
Thanks so much, guys.
Thank you.
Thank you. Bye-bye, everybody.
My pleasure.